Hello, my name is Mahima Mishra. Today we shall be discussing about the application of biotechnology in the field of medicine. One of the most important application of biotechnology in medicine field is gene therapy, which is also known as human gene transfer. So gene therapy is nothing but the replacement of faulty or missing gene in order to correct the genetic disorder. The introduction of normal gene into cells in place of missing or defective ones in order to correct genetic disorder. In medicine field, gene therapy is the therapeutic delivery of nucleic acid into a patient cell as drug to treat diseases. So the first attempt was done on a 4-year-old girl in 1990 at NIH Center, which is National Institute of Health. She was suffering from a disease in which her ADA synthesizing gene was faulty. So that disease leaves her defenseless against infection. First unsuccessful attempt was performed in 1980 by Martin Klein, but the first successful nuclear gene transfer in human was performed in 1989, which was approved by National Institute of Health. So this diagram over here shows the basic functioning of gene therapy. As you can see, we have a cell over here which is non-functioning. So a functioning gene is replaced by a non-functioning gene to make it functional, self-functioning normally. So which is also known as gene augmentation therapy. So next we will move towards the type of gene therapy. There are basically two types of gene therapy on the basis of cell being treated. There are two types: somatic gene therapy, germline gene therapy. One thing is to note that this is divided on the basis of the type of cell being treated: somatic gene therapy, germline gene therapy. And somatic gene therapy is further divided into ex vivo and in vivo. So let's get started and see what somatic gene therapy is. Basically, what somatic cell is: any cell other than germ cell is somatic cell. So somatic gene therapy, which is also known as somatic cell gene therapy, the therapeutic gene are transferred into any cell other than a gamete, germ cell or gametocyte or undifferential stem cell, SCGT, the somatic cell. So this is basically the gene therapy done in the somatic cell. The basic difference between these two is somatic cell is non-inheritable and the germline gene therapy is inheritable, because that classification has been done on the basis of the type of cell being treated. So let us see again the type of gene therapy. Two type of gene therapy: somatic gene therapy, germline gene therapy. Somatic gene therapy is gene therapy performed on a somatic cell. Somatic cell is nothing but any cell other than germ cell is called somatic cell in the body. Somatic cell is further divided into ex vivo and in vivo. So ex vivo basically means anything grown outside the living organism. That is, X stands for external. This X stands for external, and it stands for internal. So the environment, this is developed in the external environment and this is developed in the internal environment. Somatic cell gene therapy is clear. SCG is that's the basic difference I was talking about earlier. SCGT are not inheritable by offsprings because the gene therapy has been done in the somatic cell. Okay, the example is, let us see the example of somatic cell gene therapy. Introduction of gene into bone marrow, blood cell, skin cell, etc. So all these cells, that is bone marrow cell, blood cell and skin cell, all these are somatic cells. So these are the important example of somatic cell gene therapy. Now let us see the first classification of somatic gene therapy, which is ex vivo somatic cell gene therapy. Ex vivo somatic cell gene therapy is cell are modified outside the body and then transplanted back in again. Okay, so this is the first classification, and the next classification is in vivo gene therapy. In vivo gene therapy is gene are change in the cell when the cell are still in the body. That's the basic difference between these two. Ex vivo is grown outside the body, and this is inside the body. So these are the two classification which was the part of somatic cell gene therapy. Now let us move towards the germline gene therapy. In this case, cells, that is germ cells, that is cells involved in reproduction, sperm or eggs modified by the introduction of functional gene which are integrated into their genome. Okay, so basically this is nothing but gene therapy performed in the germ cell. Germ cell are those cells which are the part of reproductive system of the body. The change due to therapy would be heritable. So that's the basic difference between these two. The germline gene therapy is heritable, while the somatic cell gene therapy was non-heritable and would be passed on to the next generation. So at the end, we are going to see some of the gene therapy trials on which diseases gene therapy has been successfully performed. So these diseases are sickle cell diseases, hemophilia, mesothelioma, advanced central nervous system, ornithine transcarbamylase deficiency. So thank you. So this was all about the gene therapy. So we have covered all the important aspects of gene therapy, that is definition, first attempt of gene therapy, types of gene therapy, examples of gene therapy, and some of the examples of gene therapy trials. So thank you. This was all about gene therapy. The main aspects has been covered into this video, and if you like this video, please subscribe to my channel, and please suggest me what next topic you want me to make video for. Thank you.